Right, so we're looking at the leaf in this. So this is a big question. So they're saying plants obtain their food by the process of photosynthesis. Let's have a look at what photosynthesis is. Right, there's your chloroplast. It's got that green pigment in called chlorophyll. The sun beams down and the plant gets carbon dioxide and water and creates its own food and turns it into glucose, and there's a waste product called oxygen. Right, so what did we say this structure was? Okay, didn't you do that? Yes, you did. Glucose. Right, that is called a chloroplast, and this green pigment is called chlorophyll. That's cool. We've got that. Right. That's that done. Alright, you also need to know this as well. Should we do the word equation? No. Let's do the chemical equation. Carbon dioxide plus water gives glucose. C2, no C2, C6, H12O6 plus oxygen. That's your first step. Then all you do is add a 6 there, a 6 there, and a 6 there in front of the small molecules. So if I had to put a word equation, I would write carbon dioxide plus water gives glucose plus oxygen. There you go. Right, explain how the structure of the leaf is adapted for its role as the organ of photosynthesis. Right, first of all, what I would write is that the leaf, this is a really good question to learn actually. It teaches you everything about it. The leaf is thin and flat, so it has a short diffusion distance and a large surface area. Okay, now that's that bit done. Right, now we're looking at the, let's draw it actually. This is the upper epidermis, the waxy, well, yeah. You've got your cells here. These are transparent. This here is your waxy cuticle. Cuticle. I think that's how it's pronounced. Right, you've got a layer of transparent cells. Transparent cells. They allow light through. Then you have a layer of densely packed palisade cells. These are palisade cells. Then you have the spongy cells, and they have air gaps in to allow diffusion to take place. So you have the spongy mesophyll cells, or the spongy cells. Shall I write that again? Spongy cells. Then at the bottom you have the waxy cuticle keeping the water in, but you have here guard cells. These are called guard cells. They change shape, open up to create a little hole or a pore called a stomata. Okay, so now we've got to describe all that. Oh, you also have this structure as well, which we can't forget. You've got a vein running through. What's a good color to use for that? If I try below. Okay, you have a vein running through, and you've got, and I think it depends on the type of plant as to what sort of structure this is. So that's a vein running through. That is called the xylem and phloem. Xylem takes water up and down. Phloem takes minerals and the food that the plant has just created during the photosynthesis process. So it's taking the products of the photosynthesis process. Right now we need to write about that. So we can say you have a transparent, that means it allows light through, waxy cuticle and cells that allow light through for photosynthesis. There. Right. And it stops losing water vapor. Right. You have the palisade layer with lots of chloroplasts at the top of the leaf to allow maximum light to be captured. Okay. What else is there? Right, the spongy mesophyll layer then. The spongy, so we're just working our way down. The spongy mesophyll layer or spongy mesophyll cells has air gaps and is moist to allow gases to diffuse in and out. Yeah, so the products of photosynthesis basically, and the raw ingredients, for example CO2, and the products of photosynthesis, the O2, it can allow those to go in and out. So it can allow them to diffuse out. Yeah. So now we can say, right, the guard cells, if I can spell guard cells there, get it. The guard cells open up to create a stomata to create stoma or stomata, to allow CO2 in and O2 and water vapor out, and water vapor out. Sometimes you'll take in O2, depends. If there's no photosynthesis taking place, there's no O2 being produced, so the plant needs O2 for respiration, for aerobic respiration. And then you have the xylem and the phloem. That's still part of the structure yet. So the xylem and the phloem are used to transport water. The xylem is used to transport water and minerals around the plant, and the phloem transports the product of the photosynthesis. Right, that's that done. That's fair enough. It's good to know. This is a really good question, actually. If you can get that into your brain, that'll be useful. Right. The table lists some structures to provide a large surface area for the diffusion of substances. Complete the table by naming the organ in which each structure is found. So the spongy mesophyll is at the bottom of the leaf. The alveoli or alveolus is in the lungs. The nephron is in the kidney. And the villus or villi are in the intestine, the small intestine. Right, you have to put small intestine, not the large intestine. So the alveolus is that little cauliflower thing at the end of your lungs. The nephron is the Bowman's capsule, first coil tubule, loop of Henle, second coil tubule, and collecting duct. That structure is the nephron. And villus is in your small intestine, and it's those little projection in your intestine increases the surface area and helps diffusion. Right. What is diffusion? Let's get this right. Diffusion is the movement of particles. Let's use the word particles or molecules. Yeah, particles from an area of high concentration to an area of low concentration right through a partially permeable membrane. No, it doesn't have to be through a partially permeable membrane because it's talking about just diffusion. And remember this, it is a passive, that's a V, process. Why? It doesn't require energy like active transfer. Let me write that down. Active transfer. Okay. Right, let's move on. A nephron is involved in the removal of substances from the blood. Describe how the substances are removed. Right, this is only this is not for double science, so you can ignore this if you're not doing this. So how do you remove stuff from the blood into the nephron? It's a process called ultrafiltration. It happens, ultrafiltration occurs in the what? Glomerulus, or in the in where? In the glomerulus, yeah. Into the Bowman's capsule. I don't know if they call it renal capsule now, but let's do that. Bowman's capsule. Right. Let's keep moving on. During photosynthesis, plants can absorb CO2 through their leaves. Again, now watch this. Describe how the structure is adapted not for diffusion but to absorb something in particular, very particular. CO2. CO2. One carbon, two oxygens. This is called carbon dioxide. Okay. Right. Right. First of all, you have a. So do you know when they're talking about absorption? Say the leaf has a large surface area, is thin and flat, and is thin. This makes the diffusion gradient or diffusion distance small, making it more efficient. Yeah, makes the short diffusion distance small, making diffusion more efficient, diffusion quicker or more efficient. Okay, that's that bit done. Right, the spongy mesophyll. Right, so remember we said you've got your palisade layer at the top, that is called palisade, and you've got this layer here is called the spongy mesophyll layer. The spongy mesophyll layer has air spaces or gaps and is moist to allow diffusion of CO2 in. Right, and O2 out. And then the last bit of what is going on there? Right, and the last bit is the guard cells. The guard cells open up to create a pore or a stomata to allow CO2 into the leaf. Yeah, so it's absorbing it. That's it, that'll do it. Write a balanced equation for the gimmick. Right, okay, what did we say about this? What did we say about this? Can you remember? Let me write the word equation. Carbon dioxide. Carbon dioxide plus Barney Uber gives glucose and oxygen. So this is the word equation, but they want chemical equation. Okay, what is carbon dioxide? So we're looking at this one. CO2. What is water? H2O. 2 hydrogens, 1 oxygen. Big line, arrowhead. Right. What is glucose? C6H12O6 plus oxygen O2. Remember, oxygen runs around in pairs, as does chlorine, as does hydrogen, as does nitrogen, and so on. Right, what did we say about how to finish this off? All the big molecules, this is not the big molecule, all the small molecules, this is not a small molecule, this is a big molecule. You simply add 6, 6, 6. Now, if you've come across 666, that according to the biblical, I think it's according to the biblical, it's the sign of the beast, the Satan, but we will not worry about that. That is the balanced chemical equation. Right. Ooh dear. Estimate the total surface of this area of this leaf. They have on right, okay. So what they're saying to you is each one of these is a square, 1 square centimeter. What would I do? Right, okay, what we'll do is we'll tick off all the full ones. Full stroke, near enough full. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45, 46, 47, 48, 49, 50, 51, 52, 53, 54, 55, 56, 56. Right. Let me write that down. 56. So the yellow ones is 56. So now what I'm going to do is go into the red ones. So I'm going to pretend that that is 1. That is 2. That is 3. Oh my god. Did I just do that? That's 3. Okay, we'll live with that. 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So we'll say that's 13, and I think that's that done. Total surface area. Remember, there's the other side. So we've got, how much have we got here? We've got 9, we've got 59. and we're going to double that. This. I'd be happy with that. 56, 66, 69. 69. If I underestimated it, 1, 2. Oh, might have 1, 2. Why would that be though? Okay. So I've said 59. They're saying 60 to 68. I don't know how that works, but right. So, well, look, we'll run with ours. 59 times 2 is 118 centimeters squared. So we'll run with that. We're happy with that. Although they gave a figure between 120 and 136. I am not worried. Right. What the student has done, they've covered the surface with petroleum jelly. Basically, it's to stop diffusion of any gases if they've applied it. So A, there is no cover. Right, so this looks like as if it will be the control variable. How many? 26 minutes. Okay, let's just keep going. Right. Surface B, the lower surface is covered. So look, this is the leaf. That's the stalk. Let me just what sort of leaf is that? Let me try it again. There's the leaf. That's the vein, and these are the branches. Right, what you've got is the top has no stomata or no guard cells. And underneath you have guard cells. So they've said here the lower one's been covered, here the upper, and here both. So they've gone from none, lower, upper, both. Right, leaf C had a lower surface of 24 and lost 0.2 in 6 hours. Right, so we now need to calculate the amount of loss of mass of leaf C in grams per centimeter cubed in 1 hour. So that was in 6 hours. So you've got to be careful with this, yeah? So what we're going to do is, what are we going to do? Per centimeter cubed per hour. So we're going to do, you've lost 0.2 grams from, so look, that's grams. Per means divided by in centimeter cubed. So what's the surface area? 24. Right, centimeter cubed. So that gives us the right unit. So what is 0.2? It should equal 0.0138, or you can round it up to 0.014. They'll allow both. Right. So we're looking at the independent variable. How many variables are there? Right, there's 3 types in there. There's independent, dependent, and control. Right, independent is the one that they're changing, and it looks, can you see what's changing? Cover, lower cover, upper cover, both cover. So it's the surface area. Surface area of leaf. Identify the independent variable. Identify the independent variable. The dependent variable is the one that you are going to measure, right? And they're measuring the loss of mass. So that's quite simple. Loss of mass. Right. One variable that they should control. So this is the control variable, the one that you keep the same in order to make the experiment fair, right? Because you don't want to change too many variables. You don't want to change more than one variable because you don't know what has had that effect. So for example, if I say I want to lose weight, so then I suddenly stop eating sweets, do exercise and fast, right? So which one helped me lose weight? Was it the sweets? Was it the exercise? Or was it the fasting? So what you do is you say, okay, I'll tell you what, I'm going to do everything else the same. I'm going to control everything else. I'm going to only remove sweets. So then I know if there is a weight loss, the weight loss is due to the sweets. So what can we keep the same here? The amount of light we apply, the temperature in the room, the amount of time you keep the experiment going, the type of jelly, what else is there? The humidity makes a difference, yeah? Humidity. All right. Good. Right. Which one lost the most and which one lost the least? Right. The most. Let's look up here. Let us get rid of this. Now, here we've covered nothing. Here we've covered the bottom, right? This is the bottom where you've got the most stomata. This one here is the top, and this is both. So what's going to happen? Straight away we can tell this is going to lose the most because nothing is covered, and this is going to lose the least because everything's covered. So we can do A and D. A most, D least. D least. D least. Okay. Don't know why I said that. Right. Now the question is, which out of these two is going to lose the most? Remember we said at the bottom you've got the stomata. These are the little cells. These are the holes or the pores that the guard cells make. Now if you've got a hole, you're going to lose moisture. You're going to lose gases. So this will be the next most loss. So it will be B. No, whoa. Have I got this the right way around? Upper surface is... So the lower surface is covered. Sorry, the lower surface is covered. I'm getting my thing is mixed. Because the lower surface is covered, the stomata are covered, you're going to lose less. So it'll be C. C because the stomata are covered, and B because the upper surface is covered. Oh, right. So I've just explained it, but we need to put it in words. So we can say, there are more stomata on the bottom surface, right? And less, if none, or fewer, less on the upper surface or top surface. Right. So A, no surface covered, so maximum loss of water or mass. Next one, the top surface covered, so stomata still losing water. Right, then you can say lower surface covered, so less water lost. And the last one is both surfaces covered, so least amount of water lost. Right, you've got a plant. So you've got a plant. Is this talking about the leaf miner? Yes. Right. So what's going to happen now? So you've got a plant. You've got a little bug. It comes in, lays its eggs. Does it lay its eggs or does it actually burrow into the leaf? So look, this is the leaf and it lands there and then it burrows into the leaf. So you can't see it on the surface. Right. Adult lays eggs. Oh, okay. So it's the, yeah. So the flies come on. It's laid the eggs, but it's laid them inside. So look, you've got your palisade layer, you've got your spongy mesophyll layer, and then you've got your epidermis, upper and lower and upper epidermis. So it'll come in and lay the eggs here, right, and then it'll disappear. Skedaddle. Right. Number 3. Explain how feeding on the mesophyll, that's the middle layer. The spongy mesophyll layer is the, right, I've seen it like this. This is the mesophyll layer. This bottom bit is the spongy mesophyll layer, and this here is the palisade mesophyll layer, really technically, I think. Right? So if these guys, when they hatch, they start feeding here, what's going to happen? That's what they're saying. So the first thing you need to talk about, just talk about how will it affect tomato production. Say, look, the yield is amount how much crop you get. The yield will decrease. Done. Yeah. Why? The what do you call it? The chloroplasts will be damaged or eaten away. So what happens when there's less chloroplast? So there is less what? Photosynthesis. Okay, so that means that there's, what does photosynthesis produce? Glucose for respiration and growth. So there will be less glucose, you can even say starch, for the plant. There you go. Okay. Pesticides are no longer. All right, okay, look, this we've done loads of this. Forget that. That's to do with random mutation, DNA, da 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 da. Right, suggest one other reason why using a pesticide. Right, forget that. Not worried about that. Okay. Right, let's just quickly do. I don't know if I've done this before. Right, the release of an insect species called Trichogramma, not Bradford grammar, but Trichogramma, has been successful in controlling the leaf miner. Trichogramma feeds on the leaf miner. So, okay, it's an insect species. Name this type of pest control. It is called biological. Right, we'll finish after this. Is a 40-minute video. Right, it's biological. Remember we said bio means life. Abiotic means no life. Suggest why the release of sterile male leaf tomato leaf miner has also been successful in controlling the leaf miner. Okay. Well, yes. Okay. Right, let's just stick to the answer. Right, if it's sterile, that means there's no sperm or it's defective. No sperm produced. So there's fewer eggs are fertilized. And also the sterile, and this is important, the sterile leaf miner, the sterile male leaf miner competes for resources with other fertile male leaf miners. So if you compete in, then they don't get as much chance to mate and breed and sort of produce offspring. Right, we're going to call it quits there. We're going to try that question next.